Lecture 18, we will be looking at the evidence of evolution. In order to understand some of the evidence that we're going to be looking at today, we need to think about the age of the Earth, because a lot of the evidence that we'll be looking at is based on fossils. So fossils compared to the organisms that are on Earth today, or fossils compared to other fossils in the soils. And so the Earth is thought to be 4.5, 4.6 billion years old. That's billion with a B. And life is thought to have started on Earth 3.8 billion years old, billion years ago. And so these numbers change slightly as different research go on. It's now thought that life might be more like 4.2 billion years old instead of 3.8, but still, in the scheme of things, not too far off. And some of the evidence of how we know, how biologists know, paleontologists know that life started this far back. There are some rocks in certain parts of the world. Canada, Australia have some of the oldest rocks in the world on the surface of the Earth, and there is evidence of microscopic organisms, fossils of microscopic organisms, and this is how they know or think that life started as far back as it did. So scientists use a geologic timescale to divide the history of the Earth into eons and eras based on the evidence of biological and geographical evidence. So I'm not going to test you on these different eons and eras. If you take Geology 101, it'll basically be, you'll be learning a lot about rocks and how rocks form. Um, Geology 102 or 112. I can't remember if it's 111 and 112 or 101, 102. But the second part of geology, you learn about the geologic timescale. A large portion of the class is the geologic timescale, and it's very interesting learning these different times and the fossils and organisms that lived on Earth during those times and how they're clumped together. And so in the next lecture, we might slightly touch on some of these, but I can't remember. But these different eras and eons are clumped together based on the types of organisms that lived during those times. So if you think of dinosaurs, there is a group of time that is distinct to when dinosaurs kind of started to show up, and then when they went extinct. Large extinctions kind of, they make borders for some of these eons and eras. Uh, Evolution is linked to these geological events, changes in the environment, select for changes in body structures and different molecular structures within the cells. These changes slowly accumulate over time, producing the variety of organisms that we see today. So we saw in our last lecture that natural selection, the conditions of the environment, are going to help select for certain morphological features of organisms. And that's the same when looking at macroevolution over a long period of time. But in macroevolution, we have the accumulation of lots of different changes taking place. So many changes that we can see how different species are changing to new species, and large differences can arise between these. Evidence of evolution is easy to find. Researchers analyze fossils, the anatomy of organisms, either the anatomy of organisms that are present today on Earth, or um, fossil anatomy compared to present day, or fossil anatomy compared to other fossils. And we also, researchers look at molecular sequences. All of these will be touched on during this lecture, and how they are used. And they use these things to learn how species are related to one another. Uh, the events of evolution are studied in context with geologic timescale. So when a common ancestor between two groups of organisms was around, that would be during a certain geologic event, a certain era or eon, and on the time scale. So, what are fossils? Well, plants and animals of the past have left lots of evidence of their existence. That evidence could be the organism itself, the structures that are left behind. They could be evidence of their movements, so footprints or trails as they move across the earth. Um, the study of these fossils is called paleontology, and it is studying fossil remains or other clues to past life. Fossils are the remains of ancient organisms, and they provided some of the first evidence of evolution. A lot of these scientists, like Darwin, knew about fossils and the links between some of these characteristics of organisms in the fossils compared to on present day Earth and things like this, and helped shape the idea of present day evolution. The early evidence for evolution came from fossils and allowed scientists to compare these organisms to one another. There's lots of diversity of life, not only on the planet today, but throughout the fossil record. And a lot of these comparisons of these organisms suggests that there are common ancestors between the organisms. They share similar structures to one another, suggesting that there is a common ancestor that particular structure came from. Collections of fossils are a recording of the organisms that lived on the earth throughout history. They're also a record of the evolution that took place, how those organisms evolved to the next generation, if you will, of organisms. A fossil is evidence of an organism that is more than 10,000 years old. So that means the fossil itself has to be 10,000 years old to be considered a fossil. The oldest fossils are over 3 billion years old. Some of them are found in Canada and Australia. These areas have some of the oldest rocks on Earth, and you can find fossils of single cellular organisms. They were some of the first organisms that were on Earth 3 billion years ago. Fossils document the history of the life on Earth. Fossils allow researchers to test predictions about evolution. So these predictions could be if a fossil was found, 200 million years ago, and then another fossil was found 100 million years ago, and there are some similarities, but the predictions are that certain morphological structures evolved to new ones. And so scientists will have these predictions of how evolution might have taken place. Well, if you find a fossil that is then in the middle, 150 million years, you might be able to see if this prediction is true, or if the morphological structure is coming from something else. At the end of this lecture, we'll have an example of something like this, looking at how fish evolved to amphibians. Fossils can form in many different ways. We're going to look at 5 different ways over the next couple slides. The first one that we're going to look at is called compression. And one example of how compression can happen, an organism, in this case a leaf, falls into a river, sinks to the bottom, falls and gets stuck in the mud here. Well, then sediment is going to cover it quickly. So a big storm comes, and a lot of sediment then is flowing through the river and covers this leaf. Over time, more and more sediment will cover this leaf, creating weight on this leaf, compressing the leaf. All of that weight is going to compress the different sediment and create a sedimentary rock with the leaf in the mix, looking like this fossil looking like this. Now, when it comes to fossils, there are lots of organisms that fall to the bottom of the river. There are lots of organisms that die on land, but not all of them turn into fossils. Lots of them will decay. Animals will eat them. Fungi will break them down. And so, for an organism to become a fossil, a lot of the times its carcass, its remains, needs to be covered quickly. Because if not, the process of decaying will happen and ruin that organism. So if the covering of this sediment happens quickly, then that leaf will be preserved well. The next way of a fossil being formed is called petrification. So in this case, we're looking at an animal dies, decaying, is buried with sediment. So the animal itself could have decayed, but the bones are going to remain. And then water with minerals and sediment will cover it up, similar to compression. But instead of compressing, this bone will be replaced with minerals, with parts of rock. And so what remains over time is not necessarily the bone itself, but it will be minerals, it will be rocks that have replaced the parts of the organic matter of the skull, and it'll be rock in the form of this skull. Two other ways fossils can be made: impressions and castings. These are very similar to one another, but there is a major difference here. So the first one we'll talk about is impression. And so this is when an organism dies and is placed in a soft sediment like mud. And so the organism lays in the mud and makes an impression in the mud. As the animal decays and dies, the impression of that animal is left behind. Sediment will fill that area and turn into a rock. The top layer can erode away, leaving the impression there. It is now in stone, and it is now a fossil of let's say, an animal impression that had died there. A cast is very similar. The organism dies in a soft sediment like mud, and then when new sediment comes on top of it, fills in the impression, all of it hardens and becomes rock. If the top sediment comes loose, doesn't erode away, but comes loose, that top sediment has filled in the impression and now has the same shape, actually a negative of that shape. It has become a cast of that shape. And it has the details and shape of the organism that had died. There is an impression. When the cast is taken away, there very well could be an impression left as well. So you can have the impression and the cast in the same instance. If the cast comes loose and is moved, then you don't have the impression next to it, type of thing. Fossils can also form through um, intact preservation. So this is a more rare type of fossil, but it does exist a lot. And so organisms are preserved very suddenly in an area without oxygen, absent of oxygen. And so one case that this can happen is if the sap of a pine tree will drip and encase an insect. Over time, that sap will turn into an amber rock, and there will be an insect inside of it preserved. That lack of oxygen keeps that insect or that organism preserved and not decaying. These conditions minimize decomposition and prevent scavenging of other animals. So fossil history is very incomplete. Even though fossil evidence is very diverse, there are lots of fossils out there all over the world. It is not a complete picture. Um, One metaphor I like to use when it comes to fossils in the history of life on Earth. So let's take your life. Let's think of your life as the history of life on Earth. And the fossil record are photos of your life. So throughout your life, there are moments that are taking place, pictures being taken. Now, let's say the fossil record is a random 10 photos. So of your life, randomly, 10 photos are taken, and that is going to represent you. Those 10 photos could be, in the sense they're random, they could be at any time. You could be sleeping in one of the photos. You could be driving a car. You could be watching TV, doing things that are just you don't find very eventful. They could be pictures of you getting married, you having kids. They could be at moments that you find eventful. So what I'm getting at is fossils are these random organisms that happen to be fossilized, while so many other organisms are not fossilized, maybe because they're soft bodied and they do not fossilize well, or they were scavenged and they were other organisms ate them before they were able to become fossils. So yes, those 10 random photos are going to tell a lot about your life. You are going to be able to learn a lot about you. Are you going to be able to learn everything? No, absolutely not. There are so many moments of your life that are missed, that are not incorporated in those 10 random photos. Now, I threw out the number 10, but I have no idea what a real ratio here would be. 1,000 photos, 1,000,000 photos. I have no idea when it comes to fossils that exist on Earth, what has been missed. No one can answer these questions. So not only are lots of organisms unable to be fossilized, lots of body parts of organisms are not able to be fossilized. So if a mammal were to be a fossil, you would see its skeleton, but you would not see the soft body parts, the flesh, the organs. These things would decay and do not fossilize as well. So you can learn a lot about an organism by their skeleton, but you cannot learn everything. We have no idea what color dinosaurs were, because we do not have their flesh to be able to. It has not been fossilized. But there are lots of people that make educated guesses towards some of these things. Not only this, but fossils that do exist can be destroyed through erosion. So as water and wind go through the landscape, they're destroying rock. Well, fossils are in these rocks, and they are destroying these fossils as well. The movement of tectonic plates, volcanoes, lava, all these things can destroy fossils. Even though fossil evidence is incomplete, it provides scientists with lots of information. One example, the fossils that you're looking at here, these are marine fossils, animals that used to live on the bottom of the sea, but they were found in Oklahoma. So this tells us, you know, someone might have moved it here, but there are so many different marine fossils that obviously they are there naturally. So that means that at one time Oklahoma must have been underwater, and in fact it was during right after the time that Pangaea was breaking up. The Tethys Sea stretched across the Mediterranean area, across the United States, and all of this area was a shallow sea teeming with life that we can now find in fossils across America. So when looking at fossils in rock layers, we need to know how exactly they compare to one another. And so a very simple kind of way of looking at here, these rock layers that we're seeing here in the Grand Canyon, the rock layers that are towards the bottom are older than the rock layers above them. These rock layers needed to be laid down first before rocks start forming on top of. Just like bricks of a wall, you have to lay the bricks on the bottom first before you start putting bricks on top. Well, this is how rocks are formed as well. Now, in this case, the fossils that are in these rock layers, they must be the ones below. These fossils must be organisms that are much older than the ones above. And then these are older than even old. These are older than the ones that are even more higher up, more above. And so dating fossils can give clues about the timeline of life's history. Researchers use several different methods to determine age, and one of these is this, you know, relative dating, which is the layer that they are in. And so relative dating dates fossils according to the rock layer. Relative dating assumes that the rock layers below have older fossils than the newer layers, the layers above. And this is indirect, simple, and less precise, but it does give some valuable information. Now, if you know anything about rocks, you do know sometimes that rocks can be twisted on top of each other, and you can get some ripples and you can get some swirling like situations. It is not as common, but it does happen, and it could be confusing because of the movement of the rocks in the fossils. But these are unusual situations. Absolute dating dates fossils using chemistry. This is a more direct method to date the fossil. And one way to absolute date is called radiometric dating. And so radiometric dating is going to be using isotopes to ID the age of some of these fossils. We had learned about isotopes in one of the beginning lectures of our chemistry. Basically, an isotope of a chemical is it has the same number of protons, but it has different number of neutrons inside of it. So let's look at carbon. Carbon dating is talked about a lot. And so let's figure out what carbon dating is. So carbon is it has 12 protons in it. I'm sorry, it has 6 protons in its nucleus. And the most stable form of carbon is going to have 6 neutrons in its nucleus. And so that means that carbon 12, because 6 plus 6, so carbon 12 is the more stable carbon that's out there, and there's much more carbon 12 than any of the other carbons. But there is carbon 13, there is a carbon 14, and this means that they have more neutrons. Carbon 14 has 8 neutrons in it, and 6 protons still. So it has to have the 6 protons because that's what makes it carbon. And so radiometric dating is going to be measuring the amount of carbon 14 in a fossil to calculate how old that organism, or how long ago that organism died. So living organisms, they have a certain number of these isotopes inside of us. So there's a certain ratio of us that is carbon 12 and carbon 14. The world around us has a certain ratio of carbon 12 and carbon 14. And as carbon is replaced in our body, it's replaced in that same ratio. So let's say, 1% of our body is carbon 14. And so on a constant basis, as we live, that carbon 14 is going to be 1%. Every organism on Earth will have 1% carbon 14 constantly. So throughout the life, organisms are accumulating carbon 14, this radioactive isotope, and also we are replacing our carbon 12. So in this case, 99% of our carbon is carbon 12, and so that keeps replenishing itself. Same with the carbon 14. But when an organism dies, the carbon is not replenished anymore. It is what it is. And when an organism dies, it's not eating anymore. Its cells are not replacing molecules. It is just a dead organism. And so the carbon is not being added anymore. So whatever it is, it is. Well, carbon 14 decays. It is not very stable. Carbon 12 is much more stable and it will not decay over time. But carbon 14 does. And so in a certain amount of years, 5,730 years to be precise, half of that carbon will have decayed. And this is considered its half-life. So in 5,730 years, the amount of carbon 14 that was in that organism when it died, after this amount of years, half of it will be gone. In another 5,730 years, another half will be gone. So, so the first half-life, now we have 50%. The second half-life that passes by, it will half it again, which means we now have 25%. If that happens again, another 5,730 years, it will split in half again, which will be 12.5%. And so every time this amount of years passes, half of what was there will be gone. And so this is pretty precise. The amount of decaying that happens here is right on the money when it comes to time-wise. Now, this is carbon. Carbon 14 has a half-life of this. But there are other molecules that have half-lives that are much longer. Let's say 1,000,000 years or 100,000 years. We talk about carbon 14, but there are lots of other molecules that we can do this same kind of dating. And so if you take a fossil and you don't know how old it is, and you look to see how much carbon 14 is in that fossil, then you can calculate how old it is, because the assumption here is that whatever the amount of carbon 14 that's in living organisms now was in that organism when it was alive. And so this is a way of looking at how old these fossils are. Now, if a fossil is 1,000,000 years old, you might want to use another molecule other than carbon 14, because that the half-life would be so many half-lives. It would be a really small amount. So fossils dating, dated by measuring carbon-14. Since the amount of carbon-14 decreases at a known rate, scientists can measure the proportion of C14 to C12 to see how old a fossil is. In this case, we're looking at a woolly mammoth to see how long ago it died. And then this chart here is kind of showing this is when it lived, and then when it died. Every half life, it's splitting in half. So some questions to kind of test your knowledge. Which rock layer, A, B, or C, should have fossils with the most carbon-14 in them? And so well, first you want to figure out which of those are the oldest rocks. And so C is the oldest rocks. A are the youngest rocks. And so A also is the youngest fossils. The fossils in this rock layer are younger than B and C. So then when you say, well, then which ones have the most carbon-14? Younger ones or older ones? Well, since carbon-14 is going to decay over time, that means there will be less carbon-14 in the older rock layers. There will be more carbon-14 in the younger rock layers, in the younger fossils. So A should have the most carbon-14. Researchers use radioactive isotope with a 25,000 year half-life to date a fossil that is 100,000 years old. The fossil contains blank as much as the isotope did when it was a living organism. So it has a half-life of 25,000 years, and 100,000 years has passed by. So that means in the first 25,000 years, half of this isotope remains. Then we go another 25,000 years, so now we have a total of 50,000 years. Another half is gone, so that is now a quarter remaining. When another 25,000 years goes by, we are at an eighth, and so we're at a total of 75,000 years at an eighth. Another 25,000 years, we are now at 100,000 years. We are splitting that eighth in half again. It is a 16th. So in 25,000 years, in 100,000 years, which is 4 half-lives, we are going to have 16th isotope remaining. I hope that makes sense. It's a lot easier if I'm drawing these numbers on the board. So the answer is 1/16. And so these kind of questions, you definitely should know how to answer them. There may be something on the test like this. It hopefully is not in this far of depth, but you need to know how to do these calculations in order to answer a question. And so I'm thinking that these questions that are on the test will be a little simpler. We're going to now learn about biogeography, and so this incorporates fossils to kind of show the evidence of evolution, but biogeography is more than just fossils. It is looking at different species in geographical areas, different geographical locations, and it also includes time. So this geographical location over time is an important part. And if you do not know about Pangea and plate tectonics, you're going to learn a little bit about it here. In Geology 102, you'll learn a lot more about it. So Earth's geography has changed drastically over the last 200 million years ago. And so this is showing a map of the Earth and what it looked like 280 to 200 million years ago. And then it's showing how the map of Earth has changed in these different time frames. So around 150 million years, around 75 million years ago, and then what it looks like today. So how is that happening? It is happening through plate tectonics. So plate tectonic theory explains the continental movement. According to this theory, plate tectonics forces acting deep within the planet move the Earth's crust. And so there are lots of different tectonic plates that are sitting on the surface of Earth, kind of like plates, or kind of like paper floating on water. In this case, the water is magma, which is the lava underneath. Well, it's not. Lava is on the surface. Magma is underground, and it is this molten magma in the Earth's core, right outside of the Earth's core. And these plates are floating on this magma. And as different things happen, the change in temperatures are creating this movement of these plates. And that is why these continents are slowly moving over time. Continents are still moving today. We might not be able to see it, but they are still moving. Right here in the center of the Atlantic Ocean, they are separating, I can't remember exactly, a couple centimeters to a couple inches a year, something like that. They're actually calculating how, like, the separation of the plates deep in the Atlantic Ocean, so we can actually see the movements year after year. Maybe not, sitting there watching the grass grow, but you can see over years, short amount of years, actual movement taking place. So continental movements are very strong forces. They shape evolution. Not only are they creating earthquakes and volcanoes that can kill organisms, which can change the environment, but they are also moving, and so they are separating organisms or bringing them together over time. As we see, Pangaea is this supercontinent, and when it starts to split up, we're dividing the organisms that were here. They are now not intermingling with one another. They are separated from one another. As time goes on, you see how North America and South America are not touching. Well, when they finally do touch, now we have organisms that have not been intermingling ever, are now all of a sudden connected and able to spread through. And this has happened. There were lots of marsupials in South America and no marsupials in North America. When they touched, when they came into contact, the non-marsupials came down into South America, and they outcompeted most of the marsupials. There are still some marsupials living in South America today. There is only one marsupial that has come into North America and is able to live and thrive, and that is the Virginia opossum. Virginia, that is the name of it, but it is not from Virginia. It was from South America and moved in when they connected. So the distribution of particular species fossils can reveal evolutionary events. And so when we look at fossils that go across, found in both South America and in Africa, We might wonder how that happened. Um, Now, if it was a flying species, it might make sense that they migrated across the Atlantic Ocean. And that's why they're found in both Africa and South America. Well, what about organisms that can't fly, that can't swim? Well, the reason that they are found in both of these continents is because at one time they were a large continent connected to one another, and these organisms roamed across these lands freely. And then they died and fossilized, and then Pangea broke up, separated these continents, but the fossils remain on these continents. And so finding these fossils can tell us a lot about how these different land masses must have been connected to one another. So not only does are organisms affected by the breakup of these continents drifting apart, but the fossils that are from these organisms tell us about how those continents were before they started drifting. So, the location of various fossil species around 2 million years ago tells geographers, uh, well, biogeographers, that Earth's continents at the same time were arranged into a large landmass called Pangaea. And so the reason that we know this, not only animals but plants. If plants are found across continents but not found everywhere else, then it tells us that these continents could have been connected to one another. So up until now, we've talked a lot about how biologists will compare these body parts of fossils to one another, and figure out relationships between these organisms. Well, now we'll start talking about how that's done. And so, anatomical relationships between organisms can reveal common descent, that they are coming from a single ancestor, from a common ancestor. So investigators will often look at anatomical features to determine the evolutionary relationship between two organisms. Homologous structures are inherited from a common ancestor. And as we look at all of the organisms in this picture, they all have bones that are colored certain colors. And all of these animals have similar bones in their forelimbs. So the bones that are colored are all in their forelimbs. So in a human being, we see the arm. We see the different bones of the arm. The mammal here, a lion, we see that the similar bones are found in this organism. And that makes sense. We're both humans and lions are mammals. When you look at a seal, their forelimbs are a little different looking, but the bones that are underneath are the same. They have similarities to them, just as the human and the lion. Even more so, the bat. When you look at these long phalanges, these long fingers of the bat. Yes, very different, but also very similar. How the structures are created. The bones of a bird. And this is the only mammal. This is the only non-mammal here. But it too has these same similar bones that create its forelimb. And these similarities suggest that there are common ancestor between all of them. And that common ancestor will have this bone configuration. So, as people that don't study these bones and similarities, we might not think that there are lots of similarities in these, but there are. The way that they're structured, the way that they're created, and things like that. Homology demonstrates evolutionary relationship. So homologous structures can develop differently over time. As we're seeing here, the homologous structures are the forelimbs. And yes, over time, they have formed very different ways, but still very similar bone structure. Different selective pressures among these species have led to these changes from the ancestors' bones. So as these species, as these lineages have gone on through time, they are dealing with different environmental pressures. This natural selection, and lots of time has passed to create these pretty large differences. Vestigial structures are ones that have been lost, or their functions have been lost. A vestigial structure is homologous to a functional structure of another species. So when we look at the eyes of a mole, yes, they're still there. They're very tiny. They're not all that functional because a mole does not need them, does not use them. It's underground all the time. It's using other senses. So they're still there, but very small and considered vestigial. The pelvis and back limbs of a whale, of whales and dolphins and things like that. Some very small. On the outside, you can't see that they exist. But the pelvis, very small, still there. And so they're common ancestors. They are closely related to the cow. And so, the cows have back legs, back pelvises. And so these creatures, since they're swimming all the time, they're not using their back legs. And over time, they become vestigial. They're not being used. The legs of a snake. Snakes and lizards are very closely related. Lizards have lizards have legs, forelegs and back legs. Snakes do not. They have lost them. In their anatomy, sometimes some of the species you can see vestigial pelvis or limbs. Sometimes you can't. The ones that you can't, they've been fully lost. So vestigial eyes to blind moles, hind limbs in snakes, pelvises in whales. These are all examples of vestigial structures, ones that have been lost, but the small presence of some of them shows that they were there, showing that their homologous structure was there with common ancestors. So analogous structures are different than homologous structures. Homologous structures are ones that are shared among species. Analogous structures are ones that have evolved independently. So atomical structures that are analogous, if they are superficially similar but do not derive from a common ancestor. An example here, the butterfly wings, the wings of a butterfly and the wings of a bird. Yes, they are both wings and they both help fly, but the structures that make those wings are totally different. The butterfly has an exoskeleton, has scales. The bird has internal bone structure. And so earlier we looked at the wings of a bat and the wings of a bird. Those two, those wings are analogous, even though the forelimbs of those two creatures are homologous because the bone structure is there, but the wing itself is not shared by all the common ancestors, so it is analogous. It has derived independently. The bird has feathers on its wings. and it's differently structured, whereas the bat is the stretched skin. But don't get confused, the forelimbs themselves are homologous. It's the wing itself that is analogous. So analogous structures may come forth through similar environmental pressures. In the case here, we're looking at the loss of pigment in salamanders and the loss of pigment in crawfish that live in deep caves. So in caves, there is no light. They lose their eyesight over time. They lose their pigment over time. So this similarity that's taking place is still analogous. They're not related. They just happen to be in similar environmental pressures and dealing with it similarly. This is considered convergent evolution, where these similarities are arising in different organisms because of pressures, environmental pressures. And it produces similar structures in the organisms, but they do not share a lineage. The streamlined shapes of dolphins and sharks have evolved independently. The body plan of these two animals are homologous, vestigial, analogous, a product of convergent evolution, or both C and D. So I guess you have to have some background here. You have to know a little bit about dolphins and sharks. Dolphins are mammals. Sharks are fish. They're related to fish. And so this streamlined body shape is not homologous because it's not shared by the common ancestor. Vestigial doesn't really make sense. There's nothing being lost here. Analogous, yes, this streamlined body shape is analogous to one another. It's a very similar body shape, but it has arose independently from one another. A product of convergent evolution. So the pressures of living in the ocean and swimming are shared by both of these creatures. And the reason that these analogous structures, this streamlined body shape has formed is because of the pressure of living in the ocean. So both C and D are correct. When looking at the similarities of different organisms, looking and seeing how they are forming as an embryo is very insightful. Embryonic development patterns provide big clues to evolution. And so a lot of the times when you look at two creatures, two organisms forming from their embryo, they can have lots of similarities, a lot more similarities as the embryo than later as they become, as they're born and then become an adult. So anatomical similarities are often most obvious in embryos. We're looking here at the skull of the human and the skull of a chimpanzee, and looking at the similarities in the structure. So as a fetus, you can see the chimpanzee and the human skulls are very similar. As an infant, they are changed a lot more than as a fetus. You're starting to see the similarities are separating. As an adult, much different. And so the idea here is, as a fetus, these early developmental stages are older and more ingrained into the genes, and so this is why that their similarities are seen much more as a fetus in an embryo. when we look at the embryo of a fish, a mouse, and an alligator, um, as an adult, all of these have very different body shapes. But as embryos, look how similar they are. Even in this mouse situation, yes, a mouse has a tail, but it does not have this thick tail like this embryo has. And so just the similarities here, as they are developing. And so there are embryologists that study these similarities of embryos that grow, and it's very interesting to see all the similarities that are out there. Uh, so, how do similar embryos develop into such different organisms? There are something called the homeotic genes, and they provide very large changes to the embryo. So, homeotic genes control an organism's development. Small differences in this gene expression can make a big difference between, let's say, a limb creature and a limbless creature. So these single genes, or this small amount of genes, make large differences, make large changes in the growing embryo. And so if there were a mutation in some of these homeotic genes, there can be large consequences to the developing embryo. Now, it could be it could be detrimental to the life of the embryo, it could be beneficial. But if a mutation takes place in just a single homeotic gene, there can be a large difference taking place in that particular individual. And this is kind of showing how some of these big changes might have arose. The loss of legs of a snake. That it probably didn't happen over a generation, but with changes, mutations to these homeotic genes, it can be seen how easily things like this might have happened. Looking at DNA and proteins, we can see the relatedness of individuals by the similarity in the sequences. And so we've talked about this before, looking at the similarity of individuals and what that means. If they're related to one another, their sequences of DNA are going to be much more similar. Well, it's also taking place in different species. So two different species that are more related to one another are going to have much more similarities in their DNA sequence, or in particular protein sequences, the amino acids. There could be much more similarities seen there. It is highly unlikely that two unrelated species would evolve precisely the same DNA or protein sequence by chance. It is more likely that the similarities were inherited from a common ancestor and that differences arose by mutation after the species diverged from its ancestral type. The molecular clock. So this is thought that DNA mutations are acquired at a regular rate. The number of mutations becomes this clock, and it can tell us how much time has passed since two species shared a common ancestor. Now, you might be thinking, mutations are taking place, and if a mutation is bad, then it's going to be detrimental to the organism, and that organism will not be able to share its DNA later down the line. And this is true, but most of the DNA in an organism is not part of genes. Most of the DNA probably over 90% of DNA in all organisms is not creating proteins. And so if there is a mutation taking place in that DNA, well, it's not detrimental, it's not positive, it's just a change that takes place. And it doesn't hurt or help the organism. And so it just keeps going and keeps going. And so when looking at certain parts of DNA and you're seeing the differences, you can tell how much time has passed by how many mutations you're seeing. When looking at the spread of a virus, this is one way how people can tell if a virus has come from a particular country, a particular population. You're comparing the different mutations that are found in that virus's DNA, and you're able to see, you can kind of backtrack where that virus is coming from, how long ago, things like this are looking at this molecular clock. So we talked about homologous structures like forelimbs and things like that. Homologous structures can also be molecules, can be proteins. And so, if two organisms are related to one another closely, they're going to share a lot of the same proteins. A lot of the same genes are going to be making these proteins. And so the protein sequence, the amino acids that make these proteins, will be very similar to related organisms. And as those organisms diverge with time, those sequences can change because of mutations. So when looking at the cytochrome C that is found in the mitochondria of eukaryotes, all eukaryotes have this protein. When we look at the similarities and differences between this protein, we can look and see how far apart these organisms are related. So the more amino acid differences between the species, the more distant their common ancestor was. So when comparing the human to the chimpanzee, there are 0 differences, no amino acid differences in the sequence. When looking at this monkey, there is 1 amino acid difference. The rabbit, 9, and as you go down to organisms that you might assume are not as closely related to us, you would be correct. Their protein, the differences in their protein become greater and greater. Just like looking at DNA differences, the sequences in DNA, looking at the sequences in this protein is doing the same thing. So earlier, when talking about fossils, I talked about how new fossils being discovered can kind of help figure out if scientists' hypotheses are correct or incorrect. In the way they do that is if you are predicting that a certain structure might be changing in a certain way, but you're not finding the fossils, and then when a fossil is discovered, it might show that your prediction was correct or not. And this is one case that is showing this situation taking place. So in 2006, researchers discovered fossils of an intermediate form between a fish and terrestrial vertebrates. This transitional fossil is called a Tiktaalik. This is the species, the Tiktaalik. And it showed evidence for gradual evolutionary change. Before the Tiktaalik was discovered, it was predicted to exist. Researchers were searching for a fossil that was going to represent the transition from fish to amphibian. They knew when and they knew where vertebrates moved onto land, but they hadn't found the exact fossil that showed this transition taking place. And in 2006, when they found this Tiktaalik, this is when they discovered that this hypothesis was true. I mean, they had lots of evidence that it was true before the fossil was discovered, but it's just reconfirming their hypothesis.